Yeah, good evening to all. My voice is audible. Any one of you, please let me know. My voice is audible. So my voice is audible. Okay, Deepika. Okay. Thank you, Deepika, and welcome all of you. Are you getting the screen? The screen is visible. Please let me know. Are you able to see the screen? I think screen is not visible. Or is it visible? This uh, syllabus is visible to you? Anyone? No, it's not. Just no. Just give me 1 minute then. Why it is not visible? Why it's not visible? Okay. I think now it's visible. Now it's visible to all of you. Just give me 1 minute. Yeah, it's visible now. Yeah. So I think it's visible now. Okay. So how are you all? Today we have to discuss the remaining topics which are there in your class in the genetics. I'm just waiting for 1 or 2 minutes more. Today I am not feeling well. So I will try to keep only a simple topic today. Otherwise tomorrow is Saturday and Sunday and we will try to cover the remaining parts which are there in the genetics by tomorrow. Okay. So I think I should start with today. Let me tell you how much syllabus we have done till now. So Mendel's law is there. We have done these two characters also. These quantitative and multiple alleles. Then we have done regulation of gene expression, lack of lack of brain and fine structure. These things we have done yesterday. And before that day we have done cell organelles, chromosome, mitosis and meiosis. We have also done DNA structure as well as RNA structure. So replication, transcription and translation is left and then linkage is left. Linkage chromosomal abbreviations are left. So I think today I will go with this remaining part which is of transcription and translation. Okay. I will keep it very basic and very simple because some of you are totally new to genetics. So I will try to take it very simple today. Now let me show you the. So I'm just waiting for few more students. Otherwise, I will start the class because I'm not feeling well today. I will only do a little bit topic. And one more thing is that I hope all of you have submitted me your review paper. I hope so. If someone is still has not submitted the review paper, I request to submit it as soon as possible. Okay, so if you have any query, you can let me know. Otherwise, I will start the class now. Have you submitted the review paper? Everything is fine from your side? Please let me know. Yes, please let me know. Have you submitted the review paper? Yes, please I want. I just waiting for your replies. Have you submitted the review paper? You're supposed to answer me, all of you, along with the topic. I will check it on this weekend and then I will mail you back. So please let me know the topics as well and have you done it or not? Yes, my voice is audible. Please let me know. Shivangi, Deepshika, and Shweta, Urja, please reply. Bhavni, okay, okay. Urja, try to do it when you will be free. Try to complete it because I will be free on this weekend only, so I will reply you back. So, what is the status of Deepshika? Deepshika Sarkar, please let me know. Shivangi, Komal, Shweta, all of you, you must reply. Please reply. Okay. So it's HIV infection and host gene mutation among host genetic mutation among injection drug users of northern eastern states of India. Oh, very good, Komal. The topic is quite good. Yes, Pragati, please let me know the status of your review paper. Please let me know. Okay, so I suggest all of you to try to work on review paper as well along with the assignment. Now I am going to start the today's topic because you guys are not replying to me. So it is good if I will start the topic now. Okay, all of you, I am just pronouncing your name so many times, but still you are not able to reply. No worries, I will start my topic now. Okay. So today we have to start DNA replication. So basically we have already done the structure of a DNA which was given by Watson and Crick in 1953. So we all know DNA is double helical structure. We have discussed the structure of a DNA in detail also. So DNA is a double helical structure and the three components which are there, we have a ribose sugar, along with the ribose sugar we have a phosphate group, and on the one side we have nitrogenous bases. So by these bases we are able to interconnect these polynucleotide chains. So we have adenine, cytosine, guanine and thymine. In between adenine and thymine we have double bond. Then in between guanine and cytosine we have a triple bond. So this is according to the Chargaff's rule. So we have already discussed it in detail, so I will not do it now. The main thing which we have to cover today is DNA replication. So this is the basic structure of the DNA, where nucleotides in a DNA backbone are bounded together by a phosphodiester linkage between the 3' and 5' carbons. DNA molecule has a direction. Complementary strands run in the opposite direction. So one will be from 5' to 3' in this direction and on the other side the direction is on the opposite side. So what we have to discuss today is DNA replication. So it is the basically DNA replication is the basis for inheritance. It is a fundamental process which is occurring in all the cells. Please keep this thing in mind that fundamental this DNA replication is occurring in all cells for copying DNA to the transfer genetic information to the daughter cells. So what happened basically in DNA replication? The fundamental process which is occurring in all the cells for copying DNA to transfer the genetic information to the daughter cell. So basically DNA replication is the copying of the original one. Basically because we have to transfer the genetic information from one cell to another cell. So each cell must replicate its DNA before the division. We have already discussed mitosis and meiosis. In mitosis and meiosis both, there is a division. From one cell we will get the two cells. So for if one cell is divided into two cells, definitely we need to double the DNA. So for that purpose, because one DNA will go on in one cell and another will go on another cell, so that is why before replication, before division basically, when one cell is divided into two cells, it is a need that we will double the DNA so that both the new cells, both the daughter cells, they will get the equal amount of DNA. So this is basically DNA replication. So DNA replication is the fundamental process occurring in all cells for copying DNA to transfer the genetic information to the daughter cells. So the genetic information is transferred from the single cell to the divide cell, or you can say from the parent cell to the daughter cell. So DNA replication is semi-conservative in nature. Parental strands are not degraded, rather base pairing allows each strand to serve as a template for a new strand. Newly new duplex is half parent template and half new DNA. Are you getting it? Okay, so the replication is basically it is the duplication. It is the duplication, or you can say copying of the original DNA to transfer the genetic information to the daughter cells. So DNA replication is base pairing allows each strand to serve as a template for a new strand. So DNA replication is basically semi-discontinuous. Okay. So we have in DNA replication, you have to keep one thing in mind that DNA replication is semi-conservative. So we have the old double strands, and from it we will get one strand which will be old and one strand will be new. So half duplex is from the parent which will be old, and the half duplex is the new one. So we have semi-conservative nature. So one strand is old, one strand is new. So DNA replication, this is one feature, semi-conservative. Number two, semi-discontinuous. Now what is semi-discontinuous? We have two strands in the during the replication. We have the leading strand, this is the leading strand, and we have the lagging strand also. So in leading strand the synthesis is continued. But in case of lagging strand, if you will see, we have Okazaki fragment. In case of lagging strand, we have the Okazaki fragment, and these fragments are joined by DNA ligase. So there is not continuous synthesis, rather there is a breakdown synthesis. There are fragments only which are known as Okazaki fragments, and they are joined together by the DNA ligase enzyme. So this is DNA replication. It is energy of replication. Please keep these 2 points in mind, that it is semi-conservative in nature and it is semi-discontinuous. So one strand is synthesizing it continuously, rather other strand is synthesizing it partially or by the ligasing of the fragment, Okazaki fragment. So DNA replication is energy of replication. The nucleotides arrive as nucleoside triphosphates. So we already know the side basically consists of, we have nucleosides in which DNA base sugar with PPP, which is the energy for bonding. So we have another enzyme which is here, which is DNA polymerase C, which is quite helpful in the synthesis of the new fragment. So DNA polymerase is helpful in the synthesis of the new fragment. Then we have primers. So for the replication we need primers. Okay, then DNA polymerase is needed. So strand only grows in 5 prime to 3 direction. If you are able to see, the strand only grows in from 5, the continuous strand is from the 5 prime to 3 prime direction. So the template is read in the 3 prime to 5 prime direction, while the polymerization takes place in the 5 prime to 3 prime direction. So template is basically 5 prime to 3 prime. The direction is continuous in the 5 prime to 3 prime direction. So we have RNA primers also, so synthesized by primase. These serve as a starter sequence for DNA polymerase 3. Only one RNA primer required for the leading strand and RNA primers for the lagging strand depend on the number of Okazaki fragments. So this is the DNA replication steps which are there. The number one is identification of the origin of replication. After it, unwinding. Basically, what is an unwinding? When from the double-stranded DNA, we will get the single-stranded DNA template. So if you are able to see from the double-stranded DNA, we will able to unwind it. Or you can say denaturation is there. So the number two is unwinding or denaturation of the DNA to provide a single stranded DNA template, and then formation of the replication fork. Now what is basically replication fork? Where the reaction will take place. So where the double strand will get changes into single strand, this point is known as replication fork. Before it is double stranded, then it will denatured into the single stranded one. That point is known as replication fork. So formation of the replication fork, after its initiation of the DNA synthesis and elongation, primer removal and liquidation of the newly synthesized DNA segments. So reconstitution of the chromatin structure. So all these are the steps which are there in the replication. So you have to remember all these steps during the if you have to do some theory work on it, you have to keep the mind that first step is unwinding. Number 2 is formation of replication fork. Number 3 is initiation of DNA synthesis. Then primer removal is there, and at the end we will get the chromatin structure. Now these are the important enzymes which are helpful in the replication. So the very first enzyme is DNA polymerase. So definitely DNA polymerase will be helpful in the synthesis of new strand of the DNA. This polymerase is helpful. Then we have helicase. Now what helicase will do? Let me show. Yes. So this is helicase enzyme. This enzyme will help us in unwinding the DNA, because DNA is helical in structure. This helicase enzyme will help us in unwinding the DNA, which is our first step or denaturation basically. Then we have another enzyme which is topoisomerase. Now what topoisomerase will do? It will relieve torsional strain that results from helicase induced unwinding. So it will relieve the torsional strain. After it we have RNA primase. Now what is RNA primase? RNA primase initiates synthesis of RNA primers. Then we have single-stranded binding proteins which prevent the premature reannealing of the double-stranded DNA. At the end we have DNA ligase. Now what the ligase will do? As we already discussed before, they will help in the binding of the Okazaki fragments. The ligase will help us to bind or join the partial fragments which are there. They will be joined with the help of the ligase enzyme. So these seals the single stranded link between the nascent chain and the Okazaki fragments on the lagging strand. Now this is the origin of replication in case of prokaryotes. Origin of replication is also, we just write it as ORI. There is an association of sequence specific double-stranded DNA binding protein with a series of direct repeat of the DNA sequences. So in E. coli, the ORIC is bound by the protein DNA. A complex is formed A complex is formed consisting of 150 to 250 base pair of DNA and multiples of the DNA binding protein. This leads to the local denaturation and unwinding of the AT rich region. So basically we have to unwind the AT rich region. The region which will be AT rich, we have to unwind that particular region. So, unwinding of the DNA, the interaction of proteins with ORI defines the start site of replication and provides a short region of single-stranded DNA essential for the initiation of synthesis of the nascent DNA strand. So, DNA helicase allows for processive unwinding of the DNA. So this is the unwinding, that with the help of enzyme we will unwind the region which is AT rich. Then after it, the next step is the formation of the replication fork. So the polymerase III hollow enzyme, it binds to the template DNA as a part of a multiple protein complex. DNA polymerases only synthesize DNA in the 5' to 3' direction because the DNA strands, so the synthesis will always occur in, please keep this thing in mind, the synthesis will there only in the 5' to 3' direction because the DNA strands are anti-parallel, the polymerase functions asymmetrically. So on the leading strand, the DNA is synthesized continuously. On the lagging strand, the DNA is synthesized in the short fragments, the Okazaki fragments. So now let me show you here. This is a parental DNA. This is double-stranded in nature. This is dark blue. Okay, so this is double-stranded. So from where it will able to, the double-stranded DNA gets changes into single strand. This point is known as replication fork, and there are enzymes which is helicases which unwind this double-stranded DNA into the single-stranded DNA. Now the single-stranded DNA, we have to keep important things which are there. The single-stranded binding proteins, they stabilize unbound parental DNA. So we have this pink color protein. They will stabilize this single-stranded structure. After it, we have a DNA polymerase enzyme. Now what this DNA polymerase enzyme will do, if you able to see this gray one, this is also present here, So this gray one, this purish, this is only representation in this picture. So this DNA polymerase enzyme, this helps in the synthesis of the new DNA in the direction of 5' to 3'. So the leading strand is synthesizing continuously in the direction 5' to 3' with the help of an enzyme DNA polymerase. After it, this is a lagging strand. So the lagging strand, instead of consisting of the continuous DNA strand, there is only the fragment. So the lagging strand is synthesized discontinuously. Primase synthesizes short RNA primer, which is extended by DNA polymerase to form the Okazaki fragment. So we have DNA ligase enzyme which helps in attachment of these newly synthesized partial fragments. Okay, number one, hence we have this DNA polymerase which is helpful in the synthesis of fragmented DNA only. So this is RNA primer which is present, which will helpful in the synthesis of the partial fragment of the DNA. So after the DNA primer is replaced, After the RNA primer is replaced by DNA, the DNA ligase joins these Okazaki fragments. So this is the basically the overall picturization of the DNA replication. These are the 5 steps, 1, 2, 3, 4, 5. These are the 5 steps which are there in case of DNA replication. And this is your homework also. You have to draw this diagram, the diagram of DNA replication. Just 2 and 3 enzymes are most important here. Helicases, DNA polymerase, ligase, RNA enzyme is also there, primase is there. So you have to remember the name of all these enzymes and the functioning of every enzyme. Okay, so this is the main thing which is in case of DNA polymerase. So a number of different DNA polymerase molecules engage in the DNA replication. These share three important properties. Now these are the functioning or characteristics of these polymerase. They share three important properties: chain elongation, processivity, and proofreading. So chain elongation accounts for the rate at which polymerization occurs. Processivity is an expression of the number of nucleotides added to the nascent chain before the polymerase disengages from the template. Proofreading function identifies copying errors and corrects them. These are the enzymes which are useful in the step of DNA replication. We have DNA polymerase 1, polymerase 2, polymerase 3. Similarly, we have in case of eukaryotes, we have eukaryotic DNA polymerase and which have their different functioning as well. This is the steps again, initiation and elongation of DNA synthesis. But the main point remains the same, that in the one side we have the continuous strand and on another side we have the discontinuous strand. Then we have DNA isomerase. This is also the enzyme which is helpful in the DNA replication. So DNA topoisomerase, it acts by making the transit single cut in the backbone of the DNA, enabling the strand to swivel around each other to remove the buildup of twists. So basically we have, like, this is the structure. This is DNA which is double helix. We have strand separation when we will get the single stranded DNA, and here there is the supercoiling of the DNA. So topoisomerase will be helpful there only. And then proofread is there. You have to keep the basic thing in mind, that DNA replication steps which are there. Then some inhibitors of DNA replication. So you have to remember, let us summarize the replication. The first step is unwinding. The number 2 is primase, when we are synthesizing RNA primer. Then continuous synthesis which is in the leading strand. Discontinuous synthesis in the lagging strand. And synthesis will always occur in 5' to 3' direction. Please keep this thing in mind, that synthesis will always occur in 5' to 3' direction. Primers are removed, nick sealed. Proofreading by DNA polymerase organize it into a chromatin structure. So this is all the things which are there in the replication. I hope you will be able to understand the basics only. So are you able to understand the basics of DNA replication? So shall we continue transcription now? Transcription is very easy. Let me tell you. Okay. So shall we continue now? John, where are you? In the past session you are not present. Where were you in the last lecture? You were not present in the last 2 or 3 lectures, I think so. Why you are not there? Okay, okay. No worries. Please take care of yourself. I also feel that I have some fever today, but I have to take your attention also. So now we will move with transcription and translation. Uh, Now, what is transcription? Transcription is basically is a synthesis of single stranded RNA from a double stranded DNA. So, as we know, sorry, the DNA is double stranded. The DNA is double stranded. From this double stranded DNA, we will get the single stranded RNA. So, basically mRNA. So, please keep this thing in mind that transcription is the synthesis of single stranded RNA from double-stranded DNA. It produces messenger RNA. Now, what is translation? So, translation is the first stage of protein biosynthesis from RNA. In this process, the formation of polypeptides occurs by using mRNA. So, from DNA to RNA is transcription. From RNA to protein, it is known as translation, and it occurs in the ribosome. So transcription and translation, they both processes are the part of gene expression. So in eukaryotic cell, the nuclear envelope separates the transcription from the translation. So extensive RNA processing occurs in the nucleus. So RNA processing is there in the nucleus, and the translation will be there in the cytoplasm. So let me show you a transcription if you will get it. This is the double stranded DNA, the blue one. From this double stranded DNA, we are getting the single stranded RNA. Getting my point? We have done the fine structure of a gene. I think yesterday, if you remember. So this is the fine structure of mRNA. On the one side we have a poly A tail, and on the another side we have the capping. Here we have the capping also. So this is the RNA which we get. Now this RNA is consists of exons as well as introns. So when we have to pass it from the nucleus, it will be only exons, and introns will be these light pink one will be cut off. So this is the structure which we will get. We have poly A tail on one side and capping on the another side. We have already done the structure of a gene. So if you are able to get the structure of a gene, definitely you will understand now. So this is the whole transcription which is occurring inside the nucleus. From this nucleus, this mRNA will pass out, and this mRNA contains the whole genetic information or the message from the DNA. Now this mRNA is helpful for us for the synthesis of protein. Now what will happen? This mRNA will undergo some changes in the ribosomes. Ribosomes are there which are present. So this ribosomes, they will, along with tRNA and rrRNA, they will help us in the translation. So at the end we will get the protein. So if you are able to see here, these are the amino acids. So each amino acid attaches to its proper tRNA with the help of a specific enzyme and ATP, and we will get amino acyl tRNA synthesis. So these amino acids, they will bind with the tRNA, and then they will activate the further processing. So this is activated amino acids, they will bind here, and they will bind with the codons. AUG, they will bind, the codons will bind with the anticodons. This is our tRNA, this large one, and after it we will be able to grow the polypeptide chain, or this purple one is the protein which basically we get. So these proteins are synthesized by the process of translation. So these are ribosomes. We have the two subunits of ribosome. The ribosomes along with tRNA along with rRNA, they will help in the synthesis of polypeptide chain. So by translation, what we will get? We will get the polypeptide chain, or we can say protein. So let's do it now. So transcription is the synthesis of mRNA from the DNA which occur in the 5' to 3' direction. Similarly, like of replication, it is little bit same like replication. So during transcription, a DNA sequence is read by an RNA polymerase which produce a complementary and anti-parallel RNA strand. So by transcription, we will get RNA strand with the help of our enzyme RNA polymerase. So transcription is the first step leading to gene expression. As we have done gene expression, so transcription is the first step which leads to the gene expression. So the stretch of DNA transcribed into an RNA molecule is called transcription unit, which encoded at least one gene. The result of transcription is a mRNA which will then be used to create that protein by the process of translation. So from DNA we will get mRNA, and from that mRNA we will get the polypeptide or the protein. This whole step is also known as central dogma. The transcription occurs in both prokaryotes and eukaryotes, but there is difference in both pro as well as in eukaryotes. Now what happens in case of prokaryotes? Prokaryotic transcription occurring in cytoplasm, as we have seen in the picture also, in the figure also. It occurs in the cytoplasm alongside translation. Sorry, it occurs in the cytoplasm, but in case of eukaryotes, it occurs inside the nucleus basically. So in the eukaryotic transcription occurring only in the nucleus, where it is separated from the cytoplasm by the nuclear membrane. So here we have the nuclear membrane. Here we have the nuclear membrane. So transcription will be there in the nucleus and translation will be there in the cytoplasm in case of eukaryotes. So eukaryotic DNA not currently used in stores as heterochromatin around histones to form the nucleosome and must be unwound as euchromatin to be transcribed. So this is the transcription which occurs in the bacteria which is there in the cytoplasm, and this is a transcription which is occurring in the eukaryotes. We have done this transcription, we have just discussed it in the previous figure. So it occurs in the cytoplasm, it occurs in the nucleus. There is no definite phase for its occurrence. It takes place in the G1 and G2 phase of the cell cycle. So a single RNA polymerase synthesizes all three types of RNA. So only one single RNA polymerase is present, which is helpful in the synthesis of three types of RNA, which is mRNA, tRNA and rRNA. In case of this, RNA polymerase 1, 2, 3, they synthesize rRNA, mRNA and tRNA. So eukaryotic transcription is little bit more complex as compared to prokaryotic transcription. These are the three stages of transcription: initiation, elongation and termination. The number one is initiation, elongation and termination. So what is basically initiation? As we have done in case of replication also, when we have to use the enzyme, then we have to unwind the DNA. So this is the very first step. Elongation is basically when the mRNA chain will be completed and then at the end it will be terminated. So let us start with initiation. So RNA polymerase, it binds to a specific DNA region. So this triangular one is the RNA polymerase enzyme which will unwind the DNA, and after it will start the transcription. So RNA polymerase binds to the specific DNA region and initiates transcription called as promoter site. So RNA polymerase is the enzyme which is responsible for the transcription. Now this RNA polymerase which is there for unwinding of the DNA segment, it consists of 5 subunits: 2 alpha, beta, beta dash and omega subunits. So it is made up of 5 subunits, and for its proper functioning all these 5 subunits are needed. So after polymerase is bound to the promoter DNA, the two strands unwind and the enzyme starts transcribing the template strand. So the DNA will unwind and the process of transcription will start. So the position on the first synthesized base of the RNA is called start site. After it we have elongation. Elongation is basically what? RNA polymerase move along the DNA template. So RNA polymerase, this whole one is the, if you able to see this one is the RNA polymerase enzyme. So this will move along with the DNA strand. So it will move along with the DNA template and subsequently synthesize RNA chain. And it will synthesize this red color RNA chain. So it will move further and further and it will synthesize this red color chain. So DNA is unwinding ahead of the moving polymerase and the helix is reformed behind it. It unwinds 10 to 20 DNA bases at a time. So at a time only 10 to 20, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So only 10 to 20 bases are unwind at a single time. So that is why after this 20 it will move forward. So it will unwind only 10 to 20 DNA bases at a time. RNA polymerase adds nucleotides in the 5' to 3' direction. Please keep this thing in mind. The direction will be continued in the 5' to 3' direction. So the new section of RNA peels away as the double helix reforms. So this double helix will reform again, and this red mRNA will be moved out. So this is the elongation process. The polymerase will move along with the DNA and we will get the RNA strand. So this is the elongation. So and this red one is a newly made RNA. After its termination, when we will get the RNA, now what we have to do? We have to stop the reaction. So termination stops when RNA polymerase reaches a section of a DNA called terminator. So terminator sequence is basically AAU. So where we have this sequence, the reaction will be stopped. After it, the RNA will be released from the RNA chain will be released out. So next the RNA strand is released and the RNA polymerase dissociates from the DNA. So the RNA strand will go through more processing. Now RNA processing. Now what is RNA processing? As I already told you before that RNA consists of introns as well as exons. And intron is a non-coding region and exon is the coding region. So that is why we have to supply the non-coding region which is introns. So introns are removed by a process called splicing to produce a messenger RNA. So if you are able to see, this is the processing of mRNA. Exons along with introns are present. So when mRNA processing is there, first there will be addition of the cap on the start side and calling a tail at the 3' side. So there will be the addition of the cap as well as the calling a tail. And also one more thing which is there, the introns will be moved out. The introns will be spliced out. And at the end we will get the mRNA which consists of cap as well as poly-A tail. So this is the complete mRNA without the introns. So this is the process of transcription. So are you getting it or not? I think transcription is very easy. It is the processing of the mRNA only. We have already discussed it in gene interaction also. After it, we have the last step which is translation. Translation is little bit difficult, but I hope you will get it. Let me try to share. So at the end we have the last thing which is the translation. Now translation will occur in the cytoplasm only. So translation is a process in which the formation of polypeptide or protein by decoding mRNA produced in the transcription. So basically in translation what we will get? We will get the protein from the mRNA. So it occurs in the ribosomes which are present in the cytoplasm. So the reaction of translation it will take place in the cytoplasm only. It begins after mRNA enters in the cytoplasm. It uses tRNA. Please keep this thing in mind that here we have tRNA, rRNA, mRNA and ribosomes, they all will are helpful in the translation. So it uses tRNA as the interpreter of mRNA. So the phases are again same. Initiation, elongation. Now instead of termination only we have translocation and termination. Otherwise initiation, elongation and termination is there. So what is basically initiation? The initiation stage of translation brings together mRNA, tRNA bearing the first amino acid of the polypeptide. So this is the mRNA. This came from the nucleus in the cytoplasm. Now on this mRNA, tRNA will attach. And the two subunits of ribosome. So ribosome is also there. mRNA and tRNA they will form a complex. The components which are involved are the large and small subunits of ribosome. Let me show you. So this is the ribosome, this brown one. So the this red one is the mRNA, this brown one is ribosome, and we have the tRNA also. So the components involved are the large and small subunits of ribosomes, mRNA, initiator tRNA in its charged form, and three factors IF1, IF2 and IF3. So these are the factors which are also there. So the tRNA has a amino acid linked to it is termed as charged tRNA. So what is basically charged tRNA? When we have amino acids which are attached with it. So the amino acids are attached with the tRNA. It leads to the formation of charged tRNA. So this is initiation. Now we have these factors also IF1, IF2. They will bind to the 30th subunit. So these IF1 and IF2 they will bind with this smaller subunit, the 30th subunit. IF3 complex with GTP then bind to the small subunit. Now we have IF3, it binds with this GTP and then it will leave the reaction. So the sampled ribosome has two tRNA binding sites. These are A site for amino acid and P site for polypeptide. So we have two sites. We have A site, we have A site as well as we have the P site. So we have this P site and we have the A site also. So the A site is where incoming amino acid tRNA molecule bind and P site where the growing polypeptide usually forms. So on the A site the polypeptide, this is the A site on which the on which the amino acid will bind, and this is P site is the final result. The P site is the final polypeptide chain. So the A site is the acceptor site on which amino acid will bind, and on the P side we will have the polypeptide chain. So the A side is where incoming amino acid will bind, and on the P side we have the polypeptide chain. So on this P side we have the complete methionine, arginine, glutamine. Similarly, so many amino acids will join and the polypeptide chain will be released out. So one major outcome of initiation is the placement of initiator tRNA in the P site. The start codon is AUG. So please keep this thing in mind. The start codon is always be AUG. So from it only, either the mRNA will be so long, but where AUG will be present, only from that particular codon the reaction will start. That is why AUG is also known as start codon. because for that AUG we have a complementary UAG. Okay, so the start codon is AUG. For it we have a complementary UAC, not UGA. For AUG we have UAC. Against A we have E, U. So these are the complementary sequences. We have codon. You have to remember the name of codon only, which is AUG. So the smallest small ribosomal subunit attaches to the 5' end of the mRNA. So the small ribosomal subunit attaches to the 5' end of the mRNA. So ribosomal unit will bind with the, this is the 5' end. Please keep this thing in mind that the units will bind with the 5' end of the mRNA. So, a ribosomal subunit holds bind to a molecule of mRNA. In the prokaryotic cell, the mRNA binding site on this subunit recognize a specific nucleotide sequence on the mRNA just upstream of the start codon. An initiator tRNA is an anticodon. So, AUG is here and we have the anticodon which is UAC. It will pair with AUG. The result will be methionine. So we will get the methionine. So methionine is the very first amino acid and after it we have another amino acid. Now the arrival of, so this is the thing which is happening along with the smaller ribosomal subunit. Now the larger subunit will bind. Now the arrival of larger subunit completes the initiation complex. Proteins called initiation factors are required to bring all the translation components together. GDP provides the energy. So this GDP is here to provide the energy for the assembly. The initiator tRNA is the P site. A site is available to the tRNA bearing the next amino acid. So we have A site, we have the P site. So P site is basically the complete polypeptide chain only. So this is the initiation step. After it we have elongation. Now what is elongation? So this is the initiation, how from AUG we will be able to bind the things together. This is the initiation. After it we have the elongation. So please keep this step in mind, how we will able to attach mRNA with the ribosome with the tRNA and how with the help of AUG the start codon the reaction will start. Now elongation. In this amino acids are added one by one to the first amino acid as amino acid delivery. So after what it will happen? We will get the change, we will get the polypeptide chain. Okay, only one is methionine and then another amino acid will bind. So in codon recognition, the mRNA codon in the A site forms hydrogen bond with the tRNA anticodon. In peptide bond formation, the ribosome catalyzes the formation of peptide bond between the amino acids. The polypeptide extending from the P site moves to the A site attached to the new amino acid. So basically there will be only the elongation of the sequence only. Now the codons, now the amino acids will come and they will bind with the methionine basically. So the number one is codon recognition. So the anticodons of the upcoming amino acid tRNA, so they will come and they will with the complementary mRNA code in the A site. Hydrolysis of the GTP is here to increase the accuracy. So what will happen? They will bind with the they will bind with the methionine and they will form the polynucleotide chain. So we have A site, we have P site. On the P site we have the final product and from the A site we will get the amino acid tRNA base pair. Now the number 2 is peptide bond formation. So an RNA molecule of large subunit catalyzes the formation of peptide bond between the new amino acid in the A site and the carboxyl end of the growing polypeptide in the P site. This step attaches the polynucleotide to the tRNA. So similarly we have another step which is translocation. Now what is translocation? The tRNA with the polynucleotide chain in the A site is translocated to the P site. So tRNA at the P site moves to E site and leaves the ribosome. So translocation is basically when they will just replacing its own position. Getting my point? They are just moving out. So this is the translocation when the tRNA in the A side, this brown one, it takes the position of the P side, and P side will take the position of the E side, and then it will be released out. So the mRNA moves along the bound tRNA, bringing the next codon to be translated. So this will be moved out. This is only the translocation. After at the end we have the termination. For the termination we have UAA, UAG, UGA. We need these for the termination. So these protein factors, release factor interact with the stop codon and cause the release of the polypeptide chain. So when we have these codons, the reaction will be stopped. So when we have UAA, UAG, UGA, what will happen then? The polypeptide chain will be released from the tRNA and the reaction will be stopped. The ribosomes will be free, mRNA is also free, and we will get the polypeptide chain. Not chain, chain. So we will get the polypeptide chain only. So this is the termination. Yes, that is all in transcription and translation. Translation is little bit more confusing, not confusing, but it is little bit more, it takes little bit more time. Otherwise, it is also interesting. I hope all of you get it. If you are not able to get it, then no issues. I will definitely help you tomorrow also, so that I will discuss translation with me. So is it comfortable? Are you getting it, or you have some queries regarding translation? Then I will share the video which is related to translation, so that you will able to understand it properly. So is it okay for today? Please let me know. Is it okay for today? So yes, that's all for today's session. We will meet tomorrow. Till then, take care of yourself and good night to all. So is it okay? Are you getting me properly? I think that's all for today's session. If you have any other query, please let me know. If you have any another query, please let me know. Otherwise, that is all for today. If you will not get the translation, then I will let you know again. So please keep the homework ready. So you have to tell me you have to draw the diagrams of the replication, transcription, and translation. So Bhavni, is it clear to all of you? Please let me know why you are not replying. Please reply. Are you getting it? Are you able to understand it? The homework which is here, replication, the diagrams of replication, translation, and transcription. Are you able to listen to me? Please reply. बबलू के बतेगी खाता लिखा लिखा आखिरी ये नहीं खाता So please, any one of you, Pooja, please write down the syllabus, not the syllabus, but the topic, the homework which is there. I request all of you to share the topic, share the homework with all of the other students in the class. Okay. So we will meet tomorrow. Till then, take care of yourself. Good night to all, and please do your homework and please submit your assignments on time and share it with another students also. That's all for today. Even to one बरोबर गोल ये थोड़ा जोर तो देखते हैं मैं तो काफी जल्दी बन गई और बड़ा बड़ा है So yes, good night to all of you. Please share the syllabus, not the syllabus, sorry. Please share the homework with all the other students in the class. Okay, so take care. Bye bye. Hello. टमाटर ले लें गोभी ले लें गोभी है भी है तो गोभी ले लें